everybody, and welcome to today's edition of the ERS Coffee Talk. I'm Michael Kreuter, a pulmonologist from Germany dedicated to interstitial lung diseases, and I'm the current secretary of the ILD Assembly. Our topic today is the past, the present, and the future of fibrotic interstitial lung diseases. A topic of great interest and of great current advances in basic, translational, and also in clinical research, but also with a very interesting history and a huge potential for future improvements in all aspects, including therapies. I'm joined by Susanne Stowasser. She's a trained pulmonologist and head of clinical development and medical affairs, pulmonary fibrosis at Boehringer Ingelheim, and also by Karen Moor. She's a pulmonologist in training at the Erasmus ILD Center in Rotterdam, Netherlands, and she's the current early career member representative of our ILD Assembly 12. Karen, we have the past, we have the present, and we have the future.
Well, let's start with the past then. Michael, what is the reason that we don't use immunomodulatory medication in IPF anymore?
Well, Karen, I think it is my gray hair which predisposes me to speak about the past. Well, in the past, IPF was understood as an immune-mediated lung disorder involving chronic inflammation of lower airways, which then progresses to fibrosis. Already in the 50s, physicians started to treat this condition, therefore, with immunomodulatory drugs. And then there was another trial, the Iphigenia trial, which then investigated the addition of an anti-oxidative therapy, NAG, in conjunction with immunomodulatory drugs, prednisolone and azathioprine, and they showed beneficial effects of this triple therapy, which then were standard of care. However, that wasn't a placebo-controlled trial, and a US group then initiated another trial, the so-called PANTHER trial, investigating that triple therapy, NAG, azathioprine and prednisolone against placebo. And what they have shown was quite interesting, because they only did not see any beneficial effects, but actually they have seen a significant increase of overall serious events, including death. And learning from this, we stopped then the use of immune modulation in patients with IPF outside of acute exacerbations. And also what we learned from this, and also from other evidence, is that IPF is not a disease where inflammation plays a significant role. We learned that it is a disease of abnormal pulmonary fibroproliferation and disorganized matrix deposition in the face of repetitive injury to an aging alveolar epithelium that is genetically predisposed to the so-called UIP formation, and again, rather than a chronic inflammatory condition.
Michael, I think along those lines, it's important to mention as well that it was the recognition of the importance of a histologic UIP pattern underlying pathology in IPF, as described almost 25 years ago by Katzenstein and Myers, that enabled a refinement of IPF and distinction from other idiopathic forms of fibrosing interstitial lung disease. This finally enabled us to include a much more homogeneous patient population into our clinical trials and ultimately paved the way to successful clinical trials.
I completely agree with you, Susanne. And that enabled us to make many steps forward. And what you just said, since about 2000, so about 25 years ago, these clinical trials then established what we now call antifibrotic drugs, which are nowadays standard of care. But Karen, question to you. What in your eyes are the current challenges with this form of antifibrotic treatment?
Yeah, well, I think, first of all, the good news is that we finally have effective treatments for IPF. And now also even for other forms of progressive pulmonary fibrosis. Yeah, but indeed there's still many unmet needs, because the current treatments only slow down FVC decline and do not stop it. And besides that, I think we really need treatments that improve patients' quality of life. And we need better tolerable treatments, but we also need to better understand when to start, but also when to stop treatments.
Karen, you mentioned progressive pulmonary fibrosis, or also called PPF, which was included in the recent guideline update of the IPF guidelines. Michael, can you tell us a little bit more about this concept PPF? What does it mean, and how does it connect to or correlate to this ongoing discussion in the field about should we split fibrotic lung diseases or should we lump diseases altogether?
Well, with the advent of effective drugs for this mostly progressive and deadly disease, IPF, and also with the improved understanding of its pathophysiology, we as an ILD community, so meaning we all together, we started to investigate also other forms of pulmonary fibrotic disorders. And we learned that there are many patients who have a very similar disease behavior as IPF. For instance, some patients in rheumatoid arthritis-associated ILD, they look like IPF and HRCT, but also clinically they behave like IPF, or very comparable patients with fibrotic HP or unclassifiable ILD and others. So recently, as you just said, we sat together in an international guideline from ERS, ATS, ALAD, and GRS, so all over the world, and we discussed about what could be a great definition for this form of patients. And especially our aim was to take disease behavior into account, meaning to measure lung function decline, looking for a symptomatic deterioration and or increased extent of the fibrosis on HRCT. So we are lumping diseases together, looking for disease behavior, but still we are splitting diseases because we still have to look, is it a systemic disease associated ILD? Because we also have to look at the atherothoracic manifestations, but also we have, for instance, to look at antigenes which are responsible for fibrotic HP development. So we lump them together for disease behavior, but we still split because other appropriate therapies also have to be taken into account.
Susanne, do you think there's also challenges with this concept of PPF?
I do, in fact, because I think the big downside with this concept is that we have to wait until patients develop this progressive disease behavior. We have to wait until they have lost already a significant amount of their lung function. And isn't it that we have tools, or how can we predict which patients will progress rapidly, will exhibit exacerbations, or will simply be at high risk for any adverse outcomes?
I think to answer these questions, we actually really need better precision medicine for pulmonary fibrosis. And more research is definitely needed to develop prognostic and ideally, I think also predictive biomarkers. And many studies currently focus on blood biomarkers, but there's also increasing attention, I think, for other potential biomarkers, such as imaging markers or exhaled breath or genetic markers. That I think that's really important. Besides this, I think more frequent home monitoring of patients can also really help to further personalized treatments, but maybe also facilitates clinical trials. And I think when we combine all these data from various sources, we might also have a role for artificial intelligence or machine learning algorithms to really be able to predict disease behavior and also response to treatment for individual patients.
Karen, a simple question to you. In your eyes, what do you think? How will we conduct future trials in pulmonary fibrosis? And perhaps how can we improve trials for these diseases?
It's becoming more difficult maybe because new treatments have to be evaluated on top of the antifibrotic standard of care, which we discussed. So that will make it more challenging to really investigate the efficacy of new investigational treatments, especially because difference in FVC will be smaller between groups. I don't know, Susanne, do you have any new ideas on how to conduct clinical trials in the future?
Indeed, Karen, there are challenges, but I think the good news is that we and also others have just recently shown that it is possible to develop a new investigational compound on top of standard of care. However, we are in a rare disease field, and I think we have to come up with more efficient trial designs requiring less patients. This including enrichment strategies, surrogate endpoints, innovative methods which leverage historical data. And this is very close to my heart. We also need to offer more remote clinical trial components that allows to broaden access for patients to these clinical trials. And I think is the prerequisite to have more diversity, inclusion, and equity in our clinical trials in IPF.
Susanne, another question to you. What about future treatments? How will our better understanding of the pathophysiology of these diseases, and I think we learned quite a lot in the last 10, 20 years, how will that impact the development of new treatments?
This is a super exciting area. Currently, all the treatments focus on what we call one master mechanism, which is fibroblast biology, because fibroblasts are the key mediators for extracellular matrix production. However, based on recent advances in research and research technology, such as single-cell sequencing, we have learned that there are different and additional cell populations, pathologic and important cell populations involved in fibrotic remodeling. These are phenotypically altered macrophages, which exhibit a fibrotic phenotype. These are dysregulated small airway epithelial cells or alveolar progenitor cell population. They all are interlinked and interact in the fibrotic niche. And we finally have to address these cell populations, these master mechanisms all together to finally be able to intervene earlier, to intervene more effectively, and to restore hemostasis, to restore repair and functionality in pulmonary fibrosis.
Thanks, Susanne. But I think we also have to add another important aspect. We as pulmonologists, we learned quite an interesting story with lung adenocarcinoma, looking at genetic alterations there. And I think we learned there that now we have effective therapies for specific mutations and alterations. And perhaps with better understanding of target therapy, as in lung carcinoma, we may improve also outcomes in fibrotic interstitial lung diseases. Perhaps you may have heard about the so-called precision trial. That is a first step into precision medicine, also in fibrotic interstitial lung diseases. And our colleagues are there investigating the potential benefits of NAC in patients with a specific TOLP genotype. Well, that is a really interesting journey, I think. Within less than 20 years from an obsolete treatment with immunomodulatory treatments now to current antifibrotic therapies, not only for IPF, but also for other fibrotic interstitial lung diseases, which behave like IPF, and many promising ideas for the future, including new diagnostic monitoring approaches and so on, but especially now for therapies. I think that is history we are making here in medicine, especially in pulmonary medicine. And that journey, at least in my eyes, also nicely shows how much combined efforts from all different scientific sources and stakeholders, including patients, improve the landscape for subjects with pulmonary fibrosis. And for sure, in my eyes, but I'm sure that also Karen and Susanne would agree to this, and all of you listening to us today, that is a continued model for the future. So Susanne and Karen, thank you very much for this very nice and insightful coffee talk. It was a great pleasure to discuss with you. And now it's time to have a cup of coffee. Thank you very much.
Thanks, Michael.
Thanks.